So welcome everybody to Strait Science. Strait Science is an evening science series put on by UAF Northwest Campus here in Nome, and also UAF Alaska Sea Grant, and you're in the home office. And both UAF Northwest Campus and Alaska Sea Grant, we serve the Bering Strait region, and the Bering Strait region is the homeland and waters of the Yupik, Inupiaq, and St. Lawrence Island Yupik peoples. And tonight's speaker, I am going to learn a lot, and I'm very excited and thankful that Chris has joined us to talk about the Jackrabbits of the North, our Alaska hares. So Chris has been a wildlife biologist for 10 years with the Alaska Department of Fish and Game. And I had to ask, in what was the non-game program, but is now the Threatened, Endangered, and Diversity program within the Alaska Department of Fish and Game. So that's the state government. I'm a blank when it comes to Jackrabbits of the North. So I have no funny stories or anything other than I've seen one and I have lots of questions. With that, Chris, take it away. We're so glad. Oh, last but not least, anybody who's on and I can't see you right this minute, please keep your video off. That will decrease the bandwidth. I do see someone is on. So Maddie Grace, if you could just turn off your video feed, that would be down at the lower left. There you go. All right. Thank you so much. Take it away, Chris. Can't wait to learn something new.
Thank you, Gay. Let me share my screen really quick. Can everybody see it?
Yes, we can see it. Looks great.
Move a few things around. There we go. Well, thanks a lot for the introduction. Hello, my name is Chris Barger, and I'm a biologist in the TED program with Alaska Department of Fish and Game. And I've been leading a statewide study on Alaska hares over the past 4 or 5 years. I've lived in Fairbanks for over 20 years now, and I've been a biologist with the state for almost 10 years. And today I'd like to talk a little bit about my research on Alaska hares, which are also called jackrabbits. So the first time I ever ran into an Alaska hare or jackrabbit was back in 2013, and it was in the hills between Quinhagak and Platinum. And it was an amazing experience. Just getting out there, being around these things, and just truly appreciating how different they really are from snowshoe hares. But before I went out there, I thought a bunny's a bunny is a bunny. And boy, was I ever wrong. Since that trip, I have just been in awe of the species and have strived to learn more and more about them ever since. So I wanted to start by talking about the 2 different species of hares that we have here in Alaska. On the left, we have the Alaska hare, which is sometimes called the tundra hare or the jackrabbit, and then those all refer to the same species. And throughout this talk, I'll probably use the name Alaska hare, which you might see in the state hunting regulation book, or I'll probably use the word jackrabbit because that's most commonly used in Western Alaska, and that's really what I use the most. And I think it's very fitting. On the right, we have the smaller snowshoe hare. And so now let's try and take a little bit deeper dive into the natural history of these 2 species. So the first thing that people notice about jackrabbits is that they are huge. These things are enormous. They can range from 7 to 13 pounds, which makes them one of the biggest hares in the entire world. And when you compare this to the smaller snowshoe, you can see in the picture here, they just dwarf them. So a snowshoe will run about 3 to 5 pounds. So they can end up being about 2 to 3 times bigger. And here in this video, you can see the jackrabbit on the left and the snowshoe hare on the right. Where do they live? Well, jackrabbits are found in Western Alaska only. And they range from the mainland of the Alaska Peninsula down south to slightly north of Kotzebue, but not in the Brooks Range or on the North Slope. And they have a pretty limited distribution in the state. When we contrast this to snowshoe hares, they are found pretty much through the entire state and in North America. And so snowshoe hares overlap in distribution with jackrabbits for the majority of the jackrabbit range. But to the extent to which these two species compete, we really don't know yet. Another main difference between the two species is in their reproductive biology. Jackrabbits can only produce one litter of babies, otherwise known as leverets, once per year. But snowshoes, on the other hand, when presented with ideal conditions, tons of food, they can breed. And well, they can breed like rabbits. And they can have multiple litters in a year and can really increase their numbers in an area quickly. And I find in Western Alaska, these snowshoe hares tend to be congregated in dense populations and are really limited to these alder willow thickets. And Alaska hares, on the other hand, they tend to be found individually or in very small groups, and they utilize multiple habitats out there, such as the open tundra, high elevation rocky outcroppings, and alder willow thickets. So they're kind of going all over the place, and the snowshoe hares are pretty much stuck in those willow alder thickets. Currently, there's a ton of research that has been done and is being done on snowshoe hares, and we know a lot about their biology, such as population cycles, predator-prey relationships with lynx, foraging ecology, and this is just a few to name, and this is to name a few. However, almost nothing is known about just the basic biology of Alaska hares. To my knowledge, there's only been two major studies, one in the 70s and one in 2013. This is an understudied yet hunted species, which we know very little about. And the historical accounts from local residents, department staff, and field experiences from across Western Alaska suggest that the hare population is substantially below these historical records. However, we have never had any surveys of the species, and that's never been done until this project began. Given this lack of information on the species, in 2013, I teamed up with two amazing biologists. Rick Marizone on the left, who is the coordinator of the small game program with ADF&G, and Travis Booms, who is a biologist in the Threatened, Endangered, and Diversity program with ADF&G. And we sat down and we slowly devised a project on jackrabbits. And this project would not be possible without their never-ending support. So thank you. We came up with three main questions we wanted to try and answer about Alaska hares. First, how many are there? And how do we even count them? And then we could use these results to create a tool for a long-term population monitoring program. Secondly, we wanted to study, where do they even go? Do they move long distances? Do they hang out in the same areas in the winter and the summer? If we harvest a large number of these hares from a small little area, how likely is it that they can reestablish themselves? And lastly, what do they eat? And is there any differences between the years? And are they different? Do they eat different things from snowshoes? So the next question is, where do we go to find large populations of jackrabbits? And this led us on a long trail throughout Western Alaska. And at the very beginning of this project, Rick visited 9 different villages across the region. And he spent a session talking with village elders, giving public presentations. And almost everyone spoke to the decline of Alaska hares and jackrabbits in their respective region. We ended up using this momentum to secure some funding for this joint project. And since then, we have conducted 17 different surveys and capture attempts at 7 different locations throughout the range, which is shown on the map by orange circles and stars. Over the past 3 years, we have been focusing our work in the Selkirk Hills Kotzebue region in the north, the areas around Nome, and Ecook, which is located south of Dillingham. These 3 areas are depicted on the map by orange stars. To date, this immense effort has resulted in me being out in the field for at least 220 days and counting. And we have collectively covered about 10,000 miles by snow machine and have been supported by 8 different staff members across the state. Currently, we plan this winter and spring to be the last of the field work for this project. So to get back to the question of how many are there, we first have to come up with a method to count them. If we can figure out how to count them, well, then we can evaluate a long-term monitoring approach and create a tool to do that. We could estimate their population sizes. We could calculate the density of hares in an area. And we could look at the male-female ratios, and we could observe changes in these areas across the years since we're surveying them 3 to 4 years in a row. Well, there are many ways to survey animals. And moose, as an example, for moose, it's possible to fly in a small aircraft along transect lines and count the moose and create an estimate of those numbers of moose in an area. For caribou, we wait till they get in tight herds in the summer, take photographs, and use those to count them and estimate how many are out there. Well, for hares and rabbits, there are many different approaches, and we tried and failed at many of them. And one of those methods was to try and find them and count them with spotlights. Another idea was to try aerial surveys with a helicopter. Also, we tried just track surveys and pellet counts. And lastly, we found that pellet transects and analyzing the DNA on the pellet was the most reliable and effective method by far. To do this, we use snow machines to follow a transect through various habitat types. And as we're going along, we collect Alaska hare fecal pellets, droppings. And we pick those up and we label them and we record the GPS location and we take them back to the bunkhouse or our camp. And once we get back there, we thaw them out and we swab them with a toothpick. And then the DNA on the toothpick can be sequenced to determine how many unique individual jackrabbits are out there and what their sexes are. And then these transects can be repeated multiple times in a year to allow a more precise population estimate. So far, we've had a 96% success rate on analysis of these pellets. But this pellet DNA can degrade really quickly during rain events. And in the late spring, pellets melt into the snow and can disappear within a day. So it's really important that we get out there and collect the pellets in the middle of winter. Which brings up the major problem that everyone in Alaska, Western Alaska knows, the weather. And now I've been in some big storms working with seabirds in the Bering Sea before in my past, but nothing compares to the Western Alaska winters. Holy cow. It's either blowing sideways, and I had a video of that, but everyone out there already knows what that looks like. Or it's like this. Oh, the flat light. Living in Fairbanks, I thought I had seen flat light before, and I thought I had snow machined in it before. Well, nope, not at all. It turns out Western Alaska flat light, it's almost impossible to tell where you're going, how fast you're going, if you're going uphill, downhill, or off a cliff. So that was definitely one of the hardest challenges. The other challenge is it would just warm up in early March. And overnight, all the snow would disappear and melt, and the rivers would start to break open and leave you stranded 90 miles from town and try and figure out how to get home or back to town as fast as possible. But some days, some really awesome days in the spring, it is the most amazing place in the world. And I find that those are the ones I really remember the most. So what did we find so far? Here we have the preliminary summary statistics from 2018 to 2020 from just our 3 primary field sites. And in that northern Kotzebue-Sellow Hills region, we surveyed roughly 300 miles on snow machine and foot, collected 200 fecal pellets, and have identified fewer than 10 hares. Now coming down to the Nome region, we have a lot longer survey transects set up, and we have surveyed roughly 1,000 miles and collected over 1,200 pellets from Alaska hares. And we've identified fewer than 100 jackrabbits in that study area. And then this winter, we will be surveying this area 2 more times, and that'll be the end. Lastly, in the Ecology region, with the help of Paul Malone, who is our local resident who helps us out tremendously in Ecology, we have surveyed about 250 miles, collected 150 pellets, and identified fewer than 20 hares. At this time, I can't provide a density estimate in these areas, but we expect this final density estimate to be quite low. So now I'd like to shift and talk about the movement ecology aspect of this project. Essentially, where do these bunnies go? Our goal has been to determine Alaska hare seasonal and annual movements, where they go in the winter and where they go in the summer, and try and gain an understanding of their potential home range size. Is it pretty small? Is it huge? Is it 50 miles? Is it 10 miles? And then we want to compare the movements in different areas of the range. Do the ones in No move further than the ones in Ecology? Do the ones in the Silhouette Hills stay right there? In order to accomplish this, we have placed satellite collars on hares, which collect roughly 2 to 3 GPS locations a day and have an average lifespan of 1 year. This project is the first to attempt to capture and collar Alaska hares. So, in order to catch a jackrabbit, we first had to figure out what kind of food they like to eat and what kind of food to use for bait. We set out a bunch of bait piles of various types of food to try and figure out what was irresistible to jackrabbits. Turns out we couldn't find anything that they just loved, but they really like banana chips. And a big thanks to Rick for bringing those along on that trip. Also turns out they will eat fresh apples and hay and alfalfa, but they don't eat a lot of it. They just swing by and eat a few of it and wander off in the night. So we figured out what they like to eat. It's pretty simple now, right? Now all I got to do is throw that bait in a live box trap that we've seen at Walmart or any other store, and we catch a jackrabbit. Simple as that. Well, I should also mention that my background is actually with birds. I've studied birds my whole adult life, and this is the first non-bird I've ever studied. And I foolishly thought, well, birds, they just fly away from you. So you got to be really creative on how to catch a bird that's just going to fly away from you. And then I thought, well, catching a giant bunny shouldn't be that hard. You know, put a box trap down, put bait in there, and they should walk in there. Well, it turns out you can throw an apple and hay in there and banana chips, and the snowshoes just walk in there instantly. And our first try with a box trap, we quickly realized that it's not going to be as simple as just throwing a bunch of box traps around and hoping a giant jackrabbit gets in there. The other thing we figured out too is that box traps are really hard to set up in Western Alaska. So you've got constant windstorms and the box drifts, and then you have a constant windstorm and all the snow blows away from underneath your box trap. And then the next windstorm, all your bait blows away. And so when we finally got good ideal conditions, it just turned out that jackrabbits would walk around the box and drop pellets around it, but they didn't really like to go in. So after that first season in 2017 and 2018, we caught 0 jackrabbits. And I got back to Fairbanks and I went back to the drawing board. And I thought to myself, I've got a lot of birds using a bow net on the ground. Why won't that work for jackrabbits? So I spent the summer working with Travis, and we tried to figure out just how you set a bow net up in the winter. And this is what we came up with. Oh, can you hear me?
Yep, we can hear you. Somebody just muted and unmuted and muted themselves. Carry on.
And this is what we came up with. So this is a bow net. And a bow net sits on the ground right here. And the orange circles are over the hinges of the bow net. which keep it under tension. There's a string located to a release right here, and it's pretty simple. Something that I would have loved to have as a kid in my backyard. Would have been quite dangerous. But basically, you just pull the string, it releases this top hoop, it releases the tension off the spring, and this top hoop goes right to the other side and carries the net along with it. So pretty simple. All you got to do is put bait in the middle, wait for a bunny to walk into it, pull the string, and bam, we got a jackrabbit. The other part of this equation is that you can't just be sitting next to this, and then you kind of have to be hidden in a blind here in the red circle. So we run a string from the bow net all the way to the blind. We wait in the blind, wait for a bunny to show up, pull the string, and bam, we have a jackrabbit. Well, the first problem that came up is that jackrabbits are pretty much nocturnal, especially in the winter, and they are more active at night. Sometimes you can see them out in the spring and obviously in the summer when it's light year round. But in the winter months, they basically hold tight during the daylight and come out at night. So I thought to myself, how do we solve this problem? We'll use night vision. Sounds really fun. I used them before in the past on a bat project. So I borrowed a pair, and turns out it worked really well. Here's what it looks like sitting in your blind, looking through the night vision all night long. And here we can see the bow net bait pile right there, and there's a trail camera marking the other side of the bow net. I thought, man, now we're in business. Well, the first time we tried this, Travis and I were in the Sellow Hills. And the first night out, it was a horribly stormy night, but we were so excited we went out there anyways. And then the next problem really showed up. Turns out it's really, really difficult to sit in a blind in the freezing cold in a chair all night long, looking through a night vision scope, waiting for a bunny to walk up to a bait pile. And at this time, you're really close to the bait, so you have to be super quiet. You can't move around, and you just have to sit there just absolutely still. Turns out that is really difficult to do. But the strapping technique does work. In the end, we ended up catching 3 hares doing this. And this one here was caught during the daylight hours, which was amazing. And that was caught in the late spring. And here you can see the bow net closed all the way around and the net around the hare. But this technique is just extremely time intensive. You can sit in this blind for days and days and nights until you finally get a jackrabbit or Alaska hare to show up to your bait pile. And it's just not sustainable in the long run. And then at the end of this season, we knew there has to be a better way to do this. So here comes ideas from Paul Malone, who's a collaborator of ours in eCook, and Travis and myself, and we sat down and we thought really hard about it. And we thought, I bet we can make some kind of big pen trap in combination with a live box trap. And slowly but surely, we put it all together. And after multiple attempts and multiple failures, we finally devised a modified pen trap that has been working over the past year. So first we start with a 50-foot long roll of chicken wire, and we try and create roughly a 8 by 12 foot pen where we've seen some recent activity. And then this pen has 2 3-foot doors, 3 foot by 3 foot doors in it. Right here are the big openings. Now it starts to get a little complicated, I'm not gonna lie. A string is attached to the doors. Right here's a string going to the doors. And then that string is held up by a nail that's wrapped through some wire. The nail is then connected by a string down to the bar of a rat trap. And then a thin monofilament fishing line goes from the trigger of the rat trap and runs all the way across the bait pile to the opposite side of the pen. And it doesn't end there. Here you can see the bait pile in the middle, and then there's the trip line is going across the top of it. And we placed a live box trap, big middle box trap in the orange circle. and that is wired into the pen. And I should also mention that this box trap is monitored continuously by a trap transmitter. So we know exactly when the doors close, and we know exactly when something has triggered this setup. And we can do this from the comfort of the bunkhouse or from wherever we're staying, which is a definite bonus than sitting in a blind all night long, shivering, trying not to fall asleep. So you can tell at this point, we're definitely grasping for straws. And we had reached pure desperation mode. And it reminds me of one of those Rube Goldberg contraptions where you use it to catch a hare, and it takes 15 steps before it finally goes off and you catch a hare. And I do have a little video about this thing going off, and unfortunately it was too big to play in the PowerPoint. So at the end of the PowerPoint, I'm going to show a couple videos if we have time and interest. But this pen trap worked really well. And last year alone, we captured 10 Alaska hares using this modified pen trap. However, here's the big caveat. It was only 4 different jackrabbits. One hare in particular, we got up and it was caught at 1 in the morning, and we snow machine out there and we collar the jackrabbit, release the jackrabbit, snow machine back, get back to bed at like 3 in the morning, and then another trap transmitter goes off and we go back out there, and turns out it was the same jackrabbit. That jackrabbit had run down a mountain, crossed a river, went up another mountain, which was 2 straight line miles away, and got caught in a different pen trap. So it really works if there's a jackrabbit around. It almost works a little too well if you put the pen traps too close together. Now that we have a jackrabbit on our hands, what do? They're big, strong animals. Well, we get a pillowcase made out of a queen sheet or a sheet, and we cover the jackrabbit in that. And then we cut a hole just big enough for its head to fit through. And once the head is through the sheet or the pillow, we place the collar around its neck, and it is just screwed on by 2 little bolts. The excess tag of this collar is then trimmed off, and the only sample we're taking from these jackrabbits is a small amount of hair before we let them go. And this little bit of hair we send off for genetic analysis, and it tells us which individual it was, and it also tells us the sex of the individual, whether it was a male or a female. And typically, we aim to have the entire handling process and collaring process done within 2 to 3 minutes. So we're not really handling these animals very long at all before we let them go. And as soon as the collar is on the hare, it'll begin sending me 2 to 3 GPS coordinates a day, and those coordinates go right back to my computer. And these collars last between a year or 2 before they run out of battery. And once again, at the end of this presentation, I will show a couple of quick videos on collaring and releasing these hares. And just for a little size reference, I'm in the orange, I'm 6'3", and I am not a small person. So these things are enormous. So in summary, we have managed to capture 8 Alaska hares, and this just required an incredible amount of effort over the years. 2 were captured in the Selkirk Hills, 3 were in the Nome region, and 3 in the Ecology region. And here's an example of 10 months of movement data from an Alaska hare and a snowshoe hare. Using this preliminary data, we have found that they can have a home range upwards of 5,000 acres, and they can easily move 3 to 4 linear straight line miles in a matter of hours. So these things can really book it across the tundra when they want to. And like most mammals, they do tend to use their core areas within their home ranges, and they use a lot of different kinds of habitats in here. And we have not observed any movements greater than 10 miles from the capture site, but hares frequently move within their home range. And in the lower right-hand corner, we have an example of a snowshoe hare movements on the same scale. As you can see, their home range is significantly smaller. Their movements are quite diminished. This hare in 10 months only made 1 movement down a valley. and they spend most of their time in these willow alder thickets. So we can use this GPS information to create a home range map. And one example is just a kernel density estimation. And this helps us to better understand where the animal is really spending its time. And it tells us how far they move, what type of habitats they prefer, and how much land area they end up using. In this example, the pink color represents where an animal spent 50% or half of its time. The blue represents 75% of its time, and the green represents 95% of the time, or where it's almost always going to be at. These color rings are referred to as isopleths. So here we have an example of a kernel density estimate from an Alaska hare and a snowshoe hare on the same scale, with 95, 75, and 50% isopleths. Those hotter pink colors represent where the animal spends half of its time. And we found, on average, Alaska hares had a home range around 3,000 acres. And half their time was spent in just 430 acres. When you compare this to snowshoe hares, the snowshoe hares used 35 acres and spent half their time in just 6 acres. If we examine these like a little bit closer on different scales, we can see that the Alaska hares, the jackrabbits, they typically have more core areas, more of these pink areas in their home range where they spend a lot of their time, and which are highlighted by the red arrows. And then they have quite a bit more core areas compared to a snowshoe, which remains in the small little area in a thicket of alder and willows. Lastly, we hope to shed some light on the winter diet of Alaska hares. What do they eat in the winter? Is basically what we're trying to figure out. And we are planning on comparing the diet of Alaska hares throughout the range, all these 7 different areas that we've collected pellets. And we want to examine the variability, the changes in individual diet across the years. Since we have genetic markers on these pellets, we can tell if the same individual has been there for 3 or 4 years in a row, and we have his samples or her samples from those 3 or 4 different years. So we can look at the variability in an individual's winter diet across these years. And lastly, we can also look at the diet of Alaska hares and snowshoe hares where they coexist. To do this, we can use next generation sequencing, which is a fancy way of saying we can take a jackrabbit pellet or a snowshoe pellet, we can extract the DNA out of it, out of that fecal pellet. We can amplify all the DNA that's in there. We run it through a computer, and it spits out all the different kinds of plant DNA that was in that fecal pellet. So we can tell how much of that, the proportions of that plant and all the different kinds of plants that were in that fecal pellet. And now there's still a lot of analysis that needs to happen. And we hope to have that completed in the next year or two. And we still have 1 more year of field work to collect samples. So all this is still in the preliminary stage, but this is the plan. And through our time in the field, Alaska hares have been observed removing snow and open tundra habitats to access vegetation. And this strategy is not commonly seen in snowshoe hare populations that remain in the willow alders. So it's quite likely that their diets are different between the 2 species. And like in that previous picture, you can see that the Alaska hare pellet in the center here, the large one, obviously, is just a different color than the other ones. And that's kind of a common theme too, as I'm out collecting pellets. Lastly, I wanted to also mention that when this project started, there were no sport hunting bag limit seasons, season dates, or salvage requirements for Alaska hares. And since then, there have been several changes to the statewide regulations. And after the last Board of Game meeting, there are now bag limits, season dates, and salvage requirements for the entire Alaska hare range. And the majority of support for this change came from the local advisory committees out in Western Alaska. And before I wrap up, I would like to thank Travis Booms for all his help and dedication throughout this project. This project would not be possible at all without his help. I just wanted to show a quick picture of before the beginning of hare work and about a month of sleep deprivation afterwards. Thanks so much, Travis. I really appreciate it. I'd also like to thank my family that lets me go out on these wild hare chases and adventures and holds the fort down in Fairbanks, and all the folks that have helped along the way, such as my friend Caleb Wiseman, who helped me go from Kotzebue to Nome on snow machines, Patrick Jones, who's been integral since the very beginning, and Sarah Germain in Nome, who's always willing to help and has an absolutely great attitude. And last but not least, Mark Nelson, who I've drug along on a few of these adventures as well. And I'm sure there are plenty other people that have helped, and I can't do this without you. So thank you very much. And with that, thank you for listening to my presentation, and I'd be happy to take any questions. And if we have time, I'd like to show a couple videos.
All right. So so thank you, Chris. That was awesome because I knew nothing of hares, and I got lots of questions, but I'm sure everyone else does. Well, while we are thinking of your questions, and we have some questions in the chat, feel free to give the Strait Science speaker some love in the chat box. It is not easy to be a public speaker, and great job. And so the audience of Strait Science is pretty nice, or pretty revealing, one or the other. But I would recommend giving some love to Chris because that was a great presentation. And you do have some questions. I was just checking. We did have a caller on, and let me just check again. I don't see them. If the caller is on, feel free to speak up at any time. And why don't we do take the first question? So we have some. We actually have some students from Nome here on tonight to listen in. So that's exciting for all of us. And they're doing a science project on how climate change affects animal populations in Alaska. Wonderful. And their question is, what affects the jackrabbit population?
What affects the jackrabbit population? Well, there's been such little study. We don't know a whole lot about why they're declining or why there appears to be less than there used to be in the 70s and the 50s. There's a lot of things that could be contributing to that, and climate change is a great example. One thing that I'm sure you're well aware of that's been happening in Fairbanks and all over Western Alaska, and I was just in Ecook, is that freezing rain and the icing rain and these warming events we have. I understand that these just can't be helpful for a fluffy Alaska hare jackrabbit that's out there getting rained on and frozen. And it also kind of buries the tundra. On the other hand, it makes it easier to walk when compared to deep snow. So we're not really sure what the exact climate effects are doing to the species. There's still quite a bit that needs to be done, especially looking at mortality and those type of studies.
Roan or Dylan, do you guys have any other follow-up question for that answer? That was your question.
Yeah.
Okay.
Yeah. Me and so, well, the question was like, how does climate change and what affects the population?
It's a great question, and that is a great research project right there. To be honest, we don't know. That's just the honest answer. Once we get back the data on what they're eating, then we could maybe have an idea if it's affecting their diet or what kind of species they really rely on. But at this point, I would worry a little bit about icing events. However, the collared hares didn't die during that last icing event around Christmas, so they withstood that. But
that was big.
That was a big one. It was bad in Fairbanks too. So
yeah,
the whole state.
All right. So thanks, Dylan. Thanks, Roan. And we'll wait to see if you have any other questions. So moving along, when growing up in Idaho, we hunted jackrabbits, which were large, skinny, gray, big-eared rabbits.
Mhm.
Always warned about them carrying disease. Those were the ones I knew as jackrabbits. So my question is the term jackrabbit something like using seagulls for any species of gull? Good question.
Good one. No, these aren't technically jackrabbits. They're actually hares. So they're tundra hares, Alaska hare. They're a hare, not a rabbit. The jackrabbits that you see in the States, you notice those big giant ears, right? They have giant ears. These guys have evolved short little ears so they don't get frozen in the winter. Jackrabbits have big ears so they disperse the heat. Forgot where I was going with that.
Jackrabbits versus why are they called jackrabbits?
I think that the reason they're called jackrabbits, and I hear that mostly, you know, in Western Alaska where they're found, is that's what most of the local people call them. They have the little bunnies and the big bunnies. They have jackrabbits, and then they have the little bunny, the little jacks. It's just a term that I've heard a lot out there. So
Oh.
they're not a true jackrabbit, but they sure look like one. They're huge, big, long legs. They look just like a jackrabbit of the north, kind of like big puffy bodies. That's about the only difference, but they do look a lot
Okay.
alike.
All right. Hope that answers it, Jim. And then from Michelle Carson, do the spatial data indicate a preference for particular snow conditions? So drifted versus hardback versus windswept bare ground.
So we
Good
haven't
question.
looked at that yet. We're still in the preliminary stages of this project. So it'd be great to come back in a couple of years when it's all done and be able to give you the final results on what we're finding. I don't think that our GPS data is at that fine a spatial scale. We get one or 2 points a day, and sometimes we'll miss 2 or 3 days. The collaring technology for small mammals is not as great as it would be for birds. And we're remote enough that we can't use collars that use cell phone towers. So it really limits the amount of data that we get back from these collars. And when we started out, we really wanted to know, are these things moving 10 miles or are they moving 100 miles? The ones that we're catching. And so I wanted a collar that would last a year, and that really changed how many points a day I got.
And just to quickly, how do the collars come off?
Oh, they don't. They're bolted on.
Okay.
They'll stay on. Yep. Unless we catch
All
the
right.
animal again.
Okay. All right. So Carol, I don't know if Carol had a question about also the terminology used.
Mhm.
Is it accurate to call hares rabbits, or it's not accurate to call hares rabbits? Just curious about the terminology of calling Alaska hares jackrabbits, not meaning to be picky, just think that terminology matters. Maybe it is accurate to call them jackrabbits. Help.
That's good. I like that. So there's a big difference between hares and jackrabbits, or hares and rabbits. Hares give birth to young that come out fully covered. Let me go back to this presentation real quick. They come out fully covered, like right here, fully covered in hair. And they're very mobile and they take off pretty quick. Rabbits, on the other hand, give birth to young that are kind of stuck in a burrow and they have to wait for their parents to take care of them. Why do I call these jackrabbits? It's a term I picked up in Western Alaska. I've spent a lot of time in a lot of the rural communities out there, and every person I meet, I try and talk to them about Alaska hares. And if I say Alaska hare or tundra hare, the majority of people don't know what I'm talking about. They grew up calling them jackrabbits. So that's why I call them the Jackrabbits of the North, is that it's a way to reach out and communicate with those folks out in those communities and be on the same page.
All right. Well, thank you for that. And there's a lot of good kudos and comments, and people really enjoyed your talk. And then somebody says it reminds me of trying to catch Bugs Bunny and Looney Tunes. That actually came to mind as well, Linda. I was thinking like, oh gosh, this is like Wile E. Coyote trying to, it's funny. Okay, question. This is from Peter Lowy, who you waved at earlier. Question, even with the small sample size, have you seen distance differences between genders?
When we first started sampling and only getting not as many individual hares per sampling event, we were seeing a skewed towards males, more males than females. And since we've increased our sampling in Nome and increased these transects, it's getting closer to 50-50, but I want to say it's about 1.6, or 1.3 males to every 1 female. So we are seeing more males than females.
And two
And if you want
questions.
the exact numbers, I can look it up later.
Are
right.
you, sorry, just to clarify, are
No,
you
go ahead.
seeing, in terms of range, are you seeing that males or females travel further?
We don't have big enough sample sizes to really look at that. And I'm really hoping this is the last year of field effort. So we're hoping we can get a good push and get out another handful of callers to be able to look at some of those smaller questions. Because right now, with only 7 callers out, it's just not very big sample size. We can answer those basic questions of how far does this guy go or this girl go? Do they go from one side of the eastern side of the Sellout Hills to the western side of the Sellout Hills? So we're kind of at that kind of analysis stage where we need to up our sample size, and we're going to give it one more hurrah if we've got the energy
For the patients.
Oh.
Let's see. Do you know how, this will be two kind of questions because they're very similar. Do you know how long they live, and are jackrabbits longer lived than snowshoe hares?
Excellent. So we don't know how long Alaska hares live. I have collected samples from adults for 4 years in a row. So we know a minimum of 4 years in the wild. If they're anything like their counterparts, the tundra hare, the Arctic hares and tundra hares and mountain hares in other parts of the world, they'll likely live in the wild at like 6 to 8 years. A snowshoe hare, on the other hand, is at the very bottom of the food chain. And as soon as they step out of these willow alder thickets, like I'm sure most people have seen in that country, they just get eaten by somebody. So they have a pretty short lifespan. A couple-year-old snowshoe is a pretty old snowshoe.
All right. And a couple more questions. You doing okay?
Oh, great.
All right. So are there any plans to study or record data on the physiological features of Alaska hares? And additionally, was there specifically a reason to not take morphological data when you trapped an individual?
Excellent. My background is in some hormone ecology in seabirds out in the Bering Sea. So I obviously was really interested in trying to get some hormone work and some blood work on these individuals. Then after the first year of catching nothing and putting just insane amount of time into this project, we realized we just need to try and figure out how to catch one. And then when we could only catch one, we thought we don't need to handle these. We want to try and minimize the handling and the kind of stress that can cause on an animal. So we need to know where they're going. So this first step at this project, or at this species, let's just get the first one done and try and put as little stress on this animal as possible in handling. Snowshoes in particular don't enjoy handling and don't do well with long periods of it. And jackrabbits, Alaska hares are quite similar, I think. And I talked to some colleagues that work on black-tailed jackrabbits down in the States, and they had similar experiences. If the capture technique was too strenuous and the handling too long, then the animal didn't live very long. So I really wanted to do some hormone work and more physiology, and I had it all lined out. But when push come to shove and the project got going, we realized we're gonna have to drop this for the sake of the animal and for the sake of just getting some callers out. But I think there's so much in this project. It's just like the first major project to ever look at the species and how to count it and what they're doing. So, it's a great first step, and you could work on this species for 20 years, I think. So there's a lot of fun things that pop in your head of questions you want to answer as a scientist.
You have another question. Hopefully that answered that question. This question is, are the Jacks territorial?
I would answer that not like territorial, like I'd think rooster would be. You walk near a rooster and it comes and attacks you. but territorial in the fact that they have an area that is theirs. They do stay in those home ranges and those collar data. They stick in those right in those areas, and they don't venture very far from that. There are other jackrabbits, Alaska hares in those home ranges. We haven't gotten to analyze how many are male and female within that size of home range, but it's something I'd like to look at. So territorial in the sense that they do stick to a territory, and they don't, the ones that we've collared so far don't venture a way out of it. But how territorial they are towards other jackrabbits, I'm not sure. In my observations in the Selkirk Hills, they're not territorial at all with snowshoes. They will feed and sit on your bow net right next to them and around 20 other snowshoes without a problem. I think it's kind of interesting too, is that like when a fox shows up, the snowshoes scatter everywhere and take off. But when a fox shows up, the jackrabbits don't really go anywhere. They're about the same size.
The original thumper.
Yep.
The original thumper. So can you stop your screen share and people then can actually maybe get a look, unless there's another slide to bring.
Do you want me to show any videos real quick? Or no? Probably about a minute.
All right, and then we just have a couple more, like 2 more questions.
Okay, see if I can get it to work.
Actually, there may be more. So why don't you show your movies and then we'll keep going with the questions. Give people a break.
Thank you. Yeah, I tried to put these in the PowerPoint, but it kept crashing the computer. So here we have a snowshoe or an Alaska hare in a queen sheet, and we've got the collar right there, and we're getting ready to collar it. And it's just a quick little video. So Travis is cutting the hole for the snowshoe or for the Alaska hare head. So then we slide his head out through that sheet, and then we put the collar around his neck. And I have a longer one, but I tried to make them smaller.
No, and that gives you the size too.
Yeah, and
big head.
Uh-oh. Here's one, the same one being released out of the sheet. And last but not least, the Rube Goldberg trap of traps. So you can see the rat trap there, the string up to the nail, the pen doors right there being held up by the nail, and I'm going to hit the trap with a carrot to test it to make sure it works. And there you can see the trap fires, the nail comes out, these doors go down and are pinned up there. And then there's a box trap here in the corner that after about 10 minutes, the hare tries to leave the pen through that box trap.
All right. It works.
Stop sharing. It is one of those interesting things that we came up with. Okay.
You ready for a couple more questions? We really have, you have a lot of interest in your presentation. So, and the subject. You got
Great.
Time for a couple more? All right.
Oh yeah.
All right.
Excellent questions.
With the genetics, have you been able to discover anything about their mating habits, heredity / interbreeding, or relationships to other populations outside Alaska?
Another great one. So at the beginning of this project, I wanted to collect blood samples from them or a piece of tissue from them. And with that genetics, you could do a fair bit of analysis. The problem is we try and limit the handling, and we've only caught 8 of these things. So to get the sample size, we looked at pellet DNA. The pellet DNA from the hare is only on the outside of the pellet. So you wipe it off with a toothpick. That DNA is not very good. And so we have to use microsatellites, which are small little pieces of DNA, to try and figure out who that individual is. It's kind of like a crime lab and looking at small amounts of DNA in a scene. So it's the same kind of thing. Unfortunately, that DNA is not good enough to do any kind of these hereditary and close kin kind of analyses. So would love to. On the next one, it was in there as an idea, but turns out we're stuck with not great DNA off a pellet.
Well, and I think that was, for me, that was going to be my trick question, but I think it was already answered earlier, and that was in the Bering Strait region, both at St. Lawrence Island and at Diomede, hares occasionally hippity hop over
Mhm.
the ocean ice and join up either from, which way are they going? Are they going
way
east to
though?
west or
Which
west to
side?
east, or,
Yeah.
what kind of visa are they holding? So that question, I think it was answered earlier, that there really isn't any specific difference between Chukotkin hares and Seward Peninsula or Western Alaskan hares. Is that
Yeah, that's a hard
got it?
question. And none of the hares we've colored have moved that far. And I'm sure it's like a moose population where you've got 1 or 5% of the population is really going to do these long distance movements. And we just haven't caught that hare yet. And odds are not great that we really will. But
Right. Just so you know, there is genetic material sitting in the UAF Museum of the North. if
and
that helps.
I know they've been using it to answer some of
Okay.
those questions. Yep.
All right. And that was thanks to Diomede, I think,
Awesome.
provided those to the museum.
Thank you.
So here's a good gnome question. How are the ptarmigan, forget the Alaska hare, how are the ptarmigan numbers while you were out and about?
There's tons of them. Is that from Sarah? Sarah
No.
Germain?
No.
Okay. So yeah, no, we see tons of ptarmigan while we're out there. At certain times of the year. These January, February surveys, I don't see a ton of ptarmigan, but last spring they were flocked up pretty good up the Cougar Rock.
I think people were kind of worried about what happened here with the wind, the rain events,
Yeah.
the icing. Yeah, it was tough.
I
So
don't think
anyway,
it's good for any of them.
What's
Well, I don't
that?
think it's good for any of those species out there.
Right. Let's see. How fast do they move?
Well, when we tried to survey them with a helicopter, we were already getting dropped off in the woods for remote camping and hare work. And we tried to survey them with a helicopter. We finally saw one, and that thing was fast. So I would guess 35 to 40 miles an hour. That thing can outrun just about anything. And I know, yeah, they can outrun a snow machine up a mountain. Maybe in some rough tussocks they can outrun you, but I've heard a lot of stories. I would say 35, 40. They're cruising. They're so fast.
That's awesome. I was going to ask you about your snow machine, but I'll save that.
Oh, yeah.
It sounds like you've put some miles on. Are Alaska hares strict herbivores? I've watched them terrorizing snowshoe hares. And I had a question about that too, because we always think of a lot of the animals as sort of like walrus. People think that walrus just eat clams. No, they actually know how to kill and consume seals. And you have something so sweet looking as an Alaska hare. Is it got a mean streak?
I want to be surprised. These things are crazy. I've seen them like running around snowshoes a little bit, and then I've seen them both feeding at the same time on a bait pile. I know that snowshoes have been recorded on trail cameras eating carrion, like dead carcasses of things. It wouldn't shock me at all if these things did that as well, but there's no evidence one way or the other. So another one, I have no idea. Hopefully there's something neat will come out of this pellet data, and we're just starting to get results back in, or we got results back in. I've just been too busy to analyze it yet. So
all right, so maybe this question then is follow up. Hi Chris, great presentation. It was great to meet you in Nome this fall. Simone Cook. Curious if you have been able to identify any predation events of collared or uncollared hares. If so, do you know who the predators were?
So of the uncollared ones, it's so remote, it's hard for me to get out there. And so if something happens near Nome, I call the folks in Nome and beg and plead and thank them very much for going back out there and looking for it. We've never recovered any collars near Nome. I recovered a collar in the Selawik Hills, and we don't know what happened. It was just a whole collar. And then we've recovered a collar in Cook and still don't know. So yeah, it usually just takes too long to get there. So I think in the next steps for the study, we really need to hammer out mortality of the species, of what's getting them. And I'm sure that as adults, they're relatively safe from most predators. We know that golden eagles take them occasionally on the Seward Peninsula, but very rarely. But I would say as the leverets, the babies, I'm sure they're pretty vulnerable to the foxes and to all the other avian raptors out there eating them. So I don't know how the recruitment of these baby bunnies are into the population. That'd be something I'd be worried about.
All right, 2 more questions. You're doing great. So there are stories of large groups of Alaska hares moving like a herd, like the
Mhm.
hillside starts to move. Do you know anything about that?
I know about that. I have absolutely no proof of that, but I have heard these stories, and other large hare species act this way in Greenland, and then the Arctic. I've heard several people talk about this, seeing 100 hares moving together. I would love to ever see something like that, but there's nowhere that I've found that has anything remotely close to that. I've talked to people that have filled sleds. I know people that have filled sleds 20 years ago, 15 years ago with 30, 50 jackrabbits or Alaska hares, and gone back to some of these same places, and it's hard to find a couple of them. I know other people that used to fly out of Kotzebue with their cubs and fill the cub and go back to Kotz, drop them off, and then go back out and fill it back up again 20, 30 years ago. And I know people that used to shoot them on the peninsula by Kotzebue, and I've honestly never seen a track or a pellet or a sign from Kotzebue to the Selkirk Hills. So
Gotcha.
I think that used to be common. It's not common anymore.
That's pretty
It
interesting.
doesn't happen anymore.
That's pretty interesting. Good question. Thanks, Carol. What are the predators of Alaska hares other than people? Who has the speed to go get them?
Golden
I guess-
eagles. It's probably the only avian predator that can get one when they're full adults, and I don't think they take many. I think Travis would know way more than I would, but I'm only aware of a couple that have ever been recovered out of a golden eagle nest. Once they hit full maturity, within a year, they hit full maturity. There's not many things that can run one down. I'm honestly not sure, but I know that their babies would be quite vulnerable in the summer to foxes and raptors.
Right. I suppose disease. They must
Disease,
have
yeah,
mortality events at
that's-
some.
that's a whole new thing in that mortality that needs to be looked at, is disease in the species.
And last question, and then a comment. Last question is, and I have a question. Do
Mhm.
core areas exhibit specific habitat differences from non-core areas of a home range?
We haven't looked at all the core areas yet, because these callers are still bringing in data. So I kind of do these analyses and just kind of get an idea for a presentation and what's kind of going on. So we haven't looked at that specifically, but on the one I showed an example of, those core areas are like some of those higher tree line areas of willow alders that are all over the Seward Peninsula. So they are kind of sticking in some of that light, dense vegetation, and then they're coming out in the wide open tundra. They have no problem going out through openness, but those core areas are kind of hanging out in thinner, like alder willow areas. But there will be more analysis on that forthcoming. So
All right, and I'll send you all the comments in the chat. Some are lovely, and
Thank
some
you.
are also these.
Thanks,
The last
everyone.
two are just for your knowledge. 40 years ago, I hunted the Alaska hare here in Nome and saw lots along the roads at night. Not so much now.
Yeah.
And the second is in 1981, I saw planes being filled along the Noatak River near Sivasok Slough. They were everywhere that year. The next year, there were no hares anymore.
It's kind of a common theme I hear, and that I've actually seen from my first time working on them in 2013 till now. Like, I've seen that in certain areas. Yep. And we're trying to figure it out, like how many are left, and next steps will be what's getting them. Well, these are some great, amazing questions.
Well, there's a lot of interest. I think you guys have tapped on something that Western Alaska, I've seen a few when I first arrived here in Nome, but I'd say it's at least 10 years since I've seen one run in front of the car, But anyway, my question to you is, there's a lot of interest, and it's actually piquing my interest in rabbits. And actually, I didn't know that big history of being able to fill planes. I mean, it sounds like you've had a massive population decline. So, and it's hard to get them. I feel sorry. You're in your chair and you're sort of frozen on one side of your face, and it's 3 o'clock in the morning, and here, eat this carrot, please, and get in the box. But what can we do as Nome? Do we call if we see hares? Are you wanting us to call the local office here in Nome and talk to Tony Gorn or Bill Dunker or Sarah Germain? Or
I think Sarah's still on the call, so you can definitely bug Sarah about it.
Yeah, I think.
Or you can contact me through email. or give me a call as well. I always like to know where they're at, and I do keep a long database of everywhere I've seen one. Every time somebody tells me where they see one, I have an Excel sheet and it goes in there. What the folks in Nome can do. It's always good to know where the numbers are and how many people are seeing. But at the same time, I'm going out there and surveying straight from town and then going 5 different directions for hundreds of miles, or starting 100 miles out of town and doing more surveys. So I am trying to count them.
So Sarah, are you on? Are you still on? Maybe she had to bug out. So
Yep.
anyway, I think that's something that you may want to, you know, especially since you have KNOM radio and Gnome Nugget on, I would say that if people see rabbits, sorry, if they see, maybe you're interested in snowshoe hares, if they see Alaska hares, I mean, there's a lot of interest in this and lots of questions. So if they see Alaska hares, is that something you want to know about? Oh, Travis, put your, thanks, Travis.
Great.
Travis, put your email in, and maybe that's something you may want to have us all report either to you right there, or sometimes the region's pretty shy to go outside of the region with their communications, especially in some of the outlying communities that people feel more comfortable, given that we have a lot of different lifestyles that sometimes the urban centered people don't understand. So yeah, maybe
definitely reach out to Bill or Sarah at the office.
Okay, so reach out to the local Fish and Game office.
Yeah, of
Brilliant.
course. They'll pass it on to me. And I just wanted to add that a couple years ago, Rick Marizone had a poster in an online portal on Fish and Game to report these sightings, but it just wasn't really getting used, so it got taken off. And we had posters set up in local offices, and it was kind of hard to get any reports on it, because we were just trying to document if people were seeing 1 or 2 or 5, or they're seeing a lot of them, because it's honestly, I've seen 12, you know, in a little area, but it's been a long time. So just trying to find a, you know, a population where to work has been a challenge.
So here's one for you then. This is in the chat box. Last winter, I happened upon an Alaska hare atop Anvil Mountain, and over the next couple of weeks was able to take friends to the same place to see the hare. Apparently it was living up there, but seemed to emerge from basically just a tiny shelter behind a small rock jutting out of the wind-slabbed snow.
Oh, I think that I sampled that hare last year, at least the pellets from that hare. So
is that the kind of thing it would help if we pick up pellets? I mean, that's what I think. If you think about those kind of things, I think people might be interested enough to help.
Let me think about that a little bit, but it's kind of a standardized process, and they can't freeze and thaw, and it's kind of tricky. But I'm also not sure where it's going to go in the future, this being the last year, and I have all this writing to do with this. So I would have to think on that one a little bit, but I would like to think more about what the folks in Nome could do. That's a great question.
So also Bryn put in that you could call the Nome office, 443-2271.
The one thing that comes right to my mind is you can still hunt these animals. So if you or a friend, or you know someone that goes out and shoots one, harvests one, and they don't want the skeleton or the internal organs or any of that stuff, save it and bring it to Fish and Game. We'll mail it to the museum and catalog it. So always looking for more samples, or you find one that gets hit on the road. Like we have a fair number of roadkill samples in the museum when there's only like 3 roads in Western Alaska. So.
All right. Any other questions? This is your time to ask. Other than that, we'll shut her down, because I just saw the last comment, everybody, on the chat is from somebody named Lois Barger. Chris Barger is our speaker. And they say, great presentation, which it was. The kids and I are so proud of your work. Love you. Now get home and help get the kids to bed. So with that, but Lois, just know that he's got to stay on a little bit afterwards, because there's some people that want to talk to him. So just give him a couple extra minutes and he'll be on his way. So thank you all for coming. Next week, Straight Science will be on a Wednesday, and we'll be talking about the McKay's bunting on St. Matthew Island. Rachel Richardson will be the speaker. So Wednesday night. Come back, Chris. I'm sure maybe someone will want to hear about rabbits. Maybe they have a question or a sighting.
Sounds great. I'd love to. Thanks for having me, and I would love to come back after we have all this analyzed.
All right.
That was excellent. Thanks a lot.
I'll be in Nome in a couple weeks.
All right. And I'll give you my number, but we'll let everybody hop off, hippity hop off.